Hi everybody! Today we'll be talking about point mutations and the effects that they have on protein production. First things first, we need to get some terminology down. Recall that the nitrogenous bases with two ring structures, adenine and guanine, are called purines. The single ring bases, cytosine and thymine, are called pyrimidines. Okay, so what is a point mutation? A point mutation is the substitution of one nitrogenous base for another. This means that there are the same number of nitrogenous bases in the sequence, but one isn't quite right. There are two forms of this mutation. The first one is called a transition. This is the substitution of nitrogenous bases with similar structures, meaning a purine for a purine and a pyrimidine for a pyrimidine. The substitution of nitrogenous bases with different structures is called transversion. This means that a purine will be substituted with a pyrimidine and vice versa. To understand point mutations, let's demonstrate with the given DNA sequence. Keep in mind the coding strand of DNA has the same sequence and polarity as the mRNA strand after transcription, except the thymine in the coding strand is replaced with uracil in mRNA. So, the translated sequence of the original RNA, without any mutation, codes for the amino acids you see on this slide. The last two codons don't get coded into the protein, so they're doled out in this image. So the first effect of point mutations that we're going to talk about is the Missense effect. This means that the point mutation of a nitrogenous base led to a change in the amino acid that was originally supposed to be formed, which changes the protein produced. If you bring your attention to the third codon in both the original and mutation strand, you can see that a transition occurred so that the adenine was substituted for a guanine. This mutated codon sequence codes for serine instead of asparagine. Another effect caused by point mutations is the nonsense effect. This effect is caused when a codon from the original strand experiences a mutation that will lead to a stop codon being produced instead of the intended amino acid. This is a drastic change because it leads to entire chunks of amino acid missing from the protein. In this example, you can see that in the second codon, a transition occurred so that the guanine was substituted with an adenine, leading to an early stop codon. The next point mutation effect we're going to talk about is known as the silent effect. It's called the silent effect because the mutation that occurs happens to code for the same amino acid as the original RNA sequence. In the fourth codon, a transversion occurs so the uracil, previously thymine in the coding strand, is now guanine. Because it codes for the same amino acid, the mutation doesn't disrupt the protein. The final point mutation effect we're going to talk about is the read-through effect. The read-through effect occurs because the stop codon in the reading frame has a mutation that causes it to code for an amino acid. For our DNA sequence, a transversion happens at the stop codon. The mutated codon codes for a cysteine amino acid, so the stop codon is lost in the sequence and the protein continues to add amino acids. I like to think about this by imagining I'm reading a book. I want to stop at a certain page, so I bookmark it. As I'm reading, the bookmark falls out without me noticing, and I read through the point where I intended to stop. Each page in this analogy can act as the amino acid, and the bookmark is your stop codon. With that, we've discussed the different effects of point mutations. Thanks for watching.